My name is Dr. Shankari Nair, and I'm a postdoctoral fellow at the Radiation Biophysics Division in the Nuclear Medicine Department at NRF iThemba Labs. In this video, we will demonstrate the use of an automated gamma H2AX Foci assay on human peripheral blood lymphocytes for its use in biodosimetry applications. The radiation risks to personnel are strictly controlled. Ionizing radiation directly affects the structure of DNA by inducing DNA breaks, particularly double-strand breaks. One of the earliest steps in the DNA double-strand break recognition process is the phosphorylation of the histone variant, H2AX, and the recruitment of repair proteins. This video will outline the use of microscopy to score the number of gamma H2AX foci per nucleus using an automated slide scanning system and analysis platform integrating the fluorescent microscope.
Lymphocytes are isolated from whole blood. Dilute whole blood with PBS in a 1-to-1 ratio, then gently lay the diluted blood onto one volume of density gradient medium. Gradually layer the blood by holding the tube slanted at 45 degrees. Transfer the suspension to a centrifuge and spin at room temperature for 20 minutes at 900 G. After centrifugation, 4 layers will form. Carefully transfer the cloudy layer containing the lymphocytes into a new sterile conical tube. Wash the lymphocytes with PBS and count using a hemocytometer. Once the total number of lymphocytes are determined, dilute the lymphocyte suspension to a concentration of approximately 800,000 lymphocytes in a 1 milliliter of complete rPMI. The samples are now ready for irradiation.
The lymphocytes are irradiated with gamma rays from a cobalt-60 source and thereafter incubated for 1 hour at 37 degrees Celsius in a humidified 5% carbon dioxide incubator. For every irradiation condition or tube, 3 slides are prepared. Place slide into the clip holder, followed by the filter card, and finally the funnel. Secure slide clips and place into the cytocentrifuge. Add 250 microliters of the cell suspension onto each funnel and spin for 30 G for 5 minutes. The use of the cytocentrifuge is essential.